Smoky Smokes. In this video I'm going to be talking to you about transport in the phloem. We're going to start off by looking at the structure of the phloem, and then I'm going to talk to you about the mass flow hypothesis. This is the method by which scientists think that sugars are transported around the plant. So we already know that plants have a vascular system, a system by which they can carry materials around their body. We know that they have the xylem tissue that takes water and dissolved mineral ions from the roots, which they absorb from the soil, and then up through the xylem, through into the leaf, and then out through the stomata in a process known as transpiration. The second thing that they need to carry around their body is the sugars that they make in photosynthesis. So they produce photosynthesis in their leaves, in their photosynthesizing cells, in their leaves. The most rapidly photosynthesizing cells in a plant are the palisade mesophyll cells close to the top of the leaf. So these cells are rapidly producing sugars, but these aren't the only cells in the plant that need that sugar. All the cells of the plant need that sugar. So sugar is a respiratory substrate. It can be used for storage. It can be used for the production of all other molecules that the plant needs. So for instance, we know that that sugar can be used to build up polysaccharides like starch or cellulose. It can be converted into fats to produce things like the cell membrane and oils, waxes within the plant. And it can be used to be converted into amino acids. We need to add a little bit of nitrogen and maybe sulfur for that. These mineral ions they get from the soil and are transported via the xylem. These amino acids can then be used to make any proteins within the plant, and we know there are lots of different proteins in the plant. They can be used to make the plant's enzymes, the plant's hormones, and structural proteins within the plant. So this sugar needs to get everywhere. It needs to go up to the flowers to make the petals and the nectar and the reproductive organs of the plant. It needs to be able to make the fruit. We need to put the fructose into the fruit. We need to use it for respiration in all the cells, and maybe we send it down to the roots to be stored, especially if plants form tubers like carrots and potatoes and turnips and that sort of thing. So how does it get there? It has a very specialized structure called a phloem. Phloem cells are very unusual, and we're going to just first draw out the diagram of the phloem cells now, and then we'll talk about the importance of each different part. So here we can see two columns of cells flowing next to each other. Together they make up the phloem tissue. On the left, we can see the sieve tube elements. The sieve tube element cells are the cells through which the dissolved sugars actually travel. On the right, we can see the companion cells. The companion cells are there to move sugars from the place that they are made, and we call that place the source, into the sieve tube element cells. So they're involved in the process of basically loading the sieve tube element cells with sugar. The companion cells are also really important, as this is a highly active process. So these companion cells have a high metabolism. So we can see we've got lots of mitochondria inside our companion cells to provide ATP for this active loading of sugars into the sieve tube elements. When we look at the sieve tube elements, we can see that they have sieve plates. So they have ends which are broken open. They've got tiny little holes in them. These holes are called pores, and the cytoplasm is able to flow through here. So what this does is it creates a space through which the dissolved sugars can travel. So we've got this big space in the middle of the cell, so we can see that the cytoplasm and the organelles, they're pushed to the side. This creates a space for a continuous flow of dissolved sugars to move through. So they've got very little cytoplasm, very few organelles, everything's pushed to the edge. This creates more room. It's basically a hollow tube through which the sugars can travel. They also have really thick, strong walls to be able to withstand the high pressure at which the sugars are travelling. So let's just recap how the structure of the phloem is related to its function. The sieve plates have pores to allow the flow of dissolved sugars through and between the sieve tube element cells. They have very little cytoplasm and very few organelles, and these are pushed up to the edge of the cell to create a hollow space in the middle for the transport of the dissolved sugars. They have strong, thick cell walls to withstand the high pressure at which the sugar is being transported. The companion cells have many mitochondria to provide ATP for the active loading, the active transport, the movement of the sugars against their concentration gradient of the sugars into the phloem cell. The companion cells also have many ribosomes for protein synthesis to produce carrier proteins to enable sugars to be transported from the companion cell into the sieve tube element. Okay, just a few key terms. The process by which sugars are moved through the phloem is called translocation. The current accepted thinking about how sugars are moved through the phloem is called the mass flow theory. We can split this into 3 stages. The first is the loading, so the transport of sucrose into the sieve tube elements from the photosynthesizing tissue. The second is the flow of the sugar through the sieve tube elements. And then the third is the unloading, so the transfer of the sucrose from the sieve tube elements into the storage cells, or any cell which is receiving the sugar. Now, if you remember from earlier, we said the cells that produce sugar are called the source cells, and the cells that take the sugar are called the sink cells. So in translocation, sugars are always being moved from source to sink. Sugars are carried through the phloem in the form of sucrose. So when glucose is made in photosynthesis, it can be converted into many different substances, and in order to be transported in the phloem, it needs to be converted into sucrose. So let's talk through each stage in turn. So sucrose is produced by photosynthesis in the leaves, and then this is moved by facilitated diffusion into the companion cells. The companion cells actively transport protons into the spaces within the cell walls. Facilitated diffusion into the sieve tube element cells. This facilitated diffusion is via co-transport. So there is a co-transport protein in the cell membrane of the sieve tube element cells that has one binding site that will bind to a proton and another binding site that will bind to a sucrose molecule. So, in order for sucrose to travel into the sieve tube element cells, it needs to move alongside this proton. So sucrose is moved via facilitated diffusion in a co-transport protein along with this proton. This is an active process because it requires the active transport of protons into the cell wall spaces. So, now you've got loads and loads of sucrose entering our sieve tube element cells of our phloem. What's this going to do to the water potential? It causes the water potential to drop. So now you've got a lower water potential. We also know that the phloem cells are right next to the xylem cells that are carrying loads and loads of water and dissolved mineral ions. So the water potential in the xylem cells is higher than in the phloem cells. The water will move via osmosis from the xylem cells into the sieve tube element cells. The water potential is also higher in the companion cells in comparison to the phloem cells. So water will also move from the companion cells into the phloem cells. So now you've got loads and loads of water and loads of sugar in these cells. This increases the pressure. It creates a high hydrostatic pressure at the source, at the sieve tube cells at the source. This causes all of this sugar and water to move down its pressure gradient, and it causes it to move towards the sink. We call this movement of the dissolved sugars down their pressure gradient the mass flow. When the dissolved sugars arrive at the sink cells, active transport is used to actively transport them out of the sieve tube cells into the sink cells. Enters the sink cells. This lowers the water potential of the sink cells in comparison to the sieve tube element cells. So, water leaves the sieve tube element cells by osmosis and enters the sink cells. This lowers the hydrostatic pressure at the sink end of the sieve tube element. This maintains your hydrostatic gradient. This maintains the pressure gradient. So we always have a high hydrostatic pressure at the source and a low hydrostatic pressure at the sink. Because things are moving down their gradient, the actual mass flow is a passive process. It just requires active transport for the loading and the unloading. This means that the movement of sugars around the plant can be affected just like any other metabolic process. So it can be affected by temperature, it can be affected by pH, and it can be affected by metabolic poisons. So, like I said earlier on, we're pretty confident about the mass flow hypothesis as a scientific community, but there are some things that suggest that there's more going on than we really understand. So there are some things that we observe in the way plants behave that suggests that something else is going on that we haven't quite figured out yet. So for instance, one of those things is the actual existence of the sieve plates. Why are they there? They actually hinder the flow of sugars. If they weren't there at all, if the ends were just completely hollow, you would have a much readier, much easier flow of sugars through the phloem, but we don't. So why are they there? Some scientists have suggested that they serve a structural function, that they add to this strength that enables the phloem to withstand this high hydrostatic pressure that's being generated at the source end, which makes a lot of sense, but that's not confirmed. The other thing that really questions the mass flow hypothesis, and this one is very difficult to explain, is that not all of these dissolved sugars are moving at the same speed. Some of them are moving faster than others. But how can that be possible? They should really all be moving at the same speed if this theory is correct. The third is that if you look at the movement of sugars to different sinks, so to the roots or to the flowers or to the growing tips of the plant, it seems to arrive at exactly the same rate. That doesn't really make any sense because this theory relies on gradients. So we would expect the sugar to be moving fastest to wherever there is the lowest concentration of sucrose because that's where the greatest pressure gradient would be created. But that's not the case, which is very odd. So those things are really unexplained. This really tells us that in actual fact, the movement of sugars around the plant is an unexplained and unanswered question in biology, and I'm sure that more experimentation will lead us to a more fulfilling answer. But currently there are many things that do support this mass flow hypothesis. One is that we can measure this pressure that is created, so there's definitely a pressure created. We can see this because if you cut open a sieve tube, the sap spurts out at high pressure. It doesn't just dribble out, it comes out at high pressure. We also can see that there is a concentration gradient. The concentration of sucrose is higher in the leaves than it is in the sink. Also, we see the downward flow in the phloem occurring in daylight, but not at night, suggesting that it relies on this concentration gradient. so it's only occurring when the sugars are actually being produced. We also see that when you have a lot of photosynthesis occurring in the leaves and you produce a load of sucrose, just a short while later, you'll see an increase in sucrose in the phloem, demonstrating that it's being moved from the leaves into the phloem. The other thing is, if you treat phloem tissue with metabolic poisons or you inhibit oxygen getting to those tissues, then translocation stops. Also, the very existence of the companion cells tells us that whatever is going on is an active process because there are many mitochondria and it's producing loads and loads of ATP. So we've sort of got both sides of this hypothesis. Is it correct? Is it not correct? I think what we can really tell is that it's unfinished. So that's everything that you need to know about the process of translocation in the phloem. One well-known experiment that is done to demonstrate that translocation through the phloem is an active process is known as the ringing experiments. In the ringing experiments, a small section of phloem is removed from the circumference of the stem of the plant. When it's removed, what we see is the region of the stem above the missing ring of tissue tends to swell, and if you take a sample of that liquid, it's sugary. And then when you look at the non-photosynthetic material below that cut, below that tissue that's been removed, like for instance the roots, these things tend to wither and die, whereas the stuff above carries on and seems just fine. This shows us that removing the phloem causes sugars to accumulate above the ring, leading to the swelling. It shows us that an interruption of the flow of sugars to the region below the ring causes the death of tissues to this region, suggesting that they're getting them via the phloem. And it also shows that the sugars are still moving up because the stuff above the cut is perfectly fine. And these things show us that it is the phloem rather than the xylem that is responsible for the translocation of sugars. Another experiment that you might come across in your reading is known as the tracer experiment. In this case, radioactive isotopes of carbon are used for tracing the movement of sucrose in the plant. So for instance, if you use carbon-14, which is a radioactive isotope of carbon, to produce sucrose, so you give the plant exposure to radioactive carbon in carbon dioxide, it absorbs that carbon dioxide, it uses it in photosynthesis, it puts it into its sucrose molecules. You can then trace the movement of these sucrose molecules throughout the plant using autoradiography. So what you would do essentially is you would cut really, really thin sections of your plant stem and you would put them on a piece of X-ray film. The film becomes blackened where it's exposed to the radiation, which is produced by the radioactive isotope of carbon in the sugar. And then what you see is these blackened regions, they correspond to where the phloem tissue is in the stem. And this shows us that it is the phloem that is responsible for translocation. Okay, so there's been loads of information here. You need to go away, think it through, draw yourself some diagrams, and then answer some questions on this topic. Remember, you'll get very few recall questions on this. Almost all will be experimental analysis, and they will basically rely on you understanding that it's an active process and that it moves under high pressure. Well done for now.